Well, our next topic is another ethics topic. This one, food biotechnology. Is it good? Is it bad? What are the ethical considerations? Well, first we need to look at what is food biotechnology, and here's a definition. We'll say that it's food technology based on biology. Okay? And by that definition, then, we have to look at ancient food biotechnology, where microbes were used for millennia, actually, to help process and ferment and produce the foods that we're used to. So fermentation by microbes to produce cheese, beer, wine, bread, these would all be examples of ancient food biotechnology. In more modern times, we have tissue culture and we have genetic engineering then that can contribute then to the production of food. And genetic engineering is really a different process from plant and animal breeding. Genetic engineering is done in the laboratory, in vitro, that is in test tubes, as opposed to plant and animal breeding, which is done with animals and plants. So genetic engineering then, as I said, done in test tubes, involves actually manipulating DNA molecules. DNA from one species is oftentimes spliced into DNA of another species. That would be called recombinant DNA. Or sometimes it's the DNA of one species is modified in some way and put back into the same species that it came from. So genes are pulled out, they're manipulated, and they're put back in to organisms, and of course they affect development. And these organisms that grow up then with the genetically modified genes, sometimes the organisms are called genetically modified. So GMOs are genetically modified organisms. Or sometimes these organisms are termed transgenic. So you'll see all of these terms: genetic engineering, recombinant DNA, genetically modified, and transgenic. And there's a history based on research that goes into biotechnology. In 1953, the structure of DNA was discovered. Here's the book written by James Watson, who shared the Nobel Prize for figuring out that DNA was a double helix. Actually, that story is very, very interesting. I highly recommend this book, and you'll get a real idea of what science was like back in the 1950s. There were several groups of biophysicists from around the world who were interested in figuring out the structure of DNA. They had just a decade before finally gotten proof that actually it was DNA that was the genetic material. It wasn't proteins that was the genetic material. It was DNA. So then the question was, how does DNA encode all of this information about life? What is its structure? And people purposefully went out to try to figure out the structure of DNA in order to get a Nobel Prize. They figured for sure there was a Nobel Prize in it. Well, Watson and Crick were racing other people like Linus Pauling. But they didn't do any experimental data. But through connections, they got a chance to see another scientist's data while she was on vacation, and that was Rosalind Franklin, who did a beautiful X-ray crystallography work. She also was trying to figure out the structure of DNA, but these guys got to see her data without her knowing it. They looked at that picture, the aha moment went off. They went back to their lab and threw together a paper in Nature and won the Nobel Prize. And Rosalind Franklin did not get any credit. So a very interesting story, and I encourage you to read this book if you have any interest in the history of science. That was 1953. Just 20 years later, by 1973, the first gene was cloned. And of course, this was in microbes, right? And in the United States. By 1977, people were saying, is this safe? Are we going to unleash some huge mutant monsters on the world? And so a conference was held, again in California, the same state where the first recombinant DNA was made. And this conference was really important because they addressed recombinant DNA safety, regulation, risk assessment, containment, and generally scientists and the public in the United States felt safe about the decisions that were made about recombinant DNA so that the research could continue because of this conference. So this brings up the question, well, who regulates biotechnology? And we have four very powerful agencies that all are involved: the Food and Drug Administration, the USDA, the Environmental Protection Agency, and the National Institutes of Health. In fact, our regulatory agencies in the United States are powerful, they're respected, and the public generally trusts these agencies to protect their safety. That's not necessarily the case in other countries. And so in other countries, especially in Europe, public distrust of recombinant DNA arose because there was public distrust of regulatory agencies, of businesses, of scientists, of anybody who might benefit from this technology. Well, by 1990, the FDA then approved an enzyme that's used in cheese making as the first biotech product for human consumption. This wasn't labeled. In fact, traditionally, the enzyme that curdles the milk involved in the first step in cheese making, that enzyme was taken, it was scraped from the stomachs of baby calves. And of course, calves would need that enzyme to break down their mother's milk. The biotech people said, why don't we just make that enzyme in safe microbes rather than having to kill cows to get it? So they took the gene from the cows, the bovine gene, and they expressed it in microbes that were safe. And this term GRAS is an FDA term generally recognized as safe. There's many kinds of organisms. Yeast, for example, is a microorganism that we generally recognize as safe. At any rate, this enzyme is used in the vast majority of cheese in the U.S. and really globally. And there are many other products of recombinant DNA in our food from microbial genetic engineering. Other food enzymes used in bread to make sweeteners, to make other products, amino acids, peptides, flavors, etc. So all of these kinds of food ingredients may be produced by genetically engineered microorganisms, and yet these are not labeled, so people are generally unaware of it. Now, by 1994, the FDA then said, well, it's okay for you to genetically engineer plants and to sell those as food. And so the Flavor Saver tomato was the first product, and this was engineered to have better shelf life. was supposed to improve the quality. And it was voluntarily labeled. People said, this is the latest and greatest thing. It's the product of biotechnology. Unfortunately, this tomato never really made a big market splash. And so the companies that had invested in genetic engineering technology looked for other markets rather than consumer products, and what they found then were agronomic traits to focus on. And so now, instead of selling tomatoes with improved quality, they would sell seed to farmers, and the seed would produce better crops. And so we have Bt corn, we have Roundup Ready soy, we have disease resistance traits, and there are many other kinds of genetically engineered traits in plants. There are other food quality traits, nutrition, metabolic products, vaccines, and we'll talk about some of these. Bt corn is something we're very familiar with here in Iowa. It contains a gene that produces a natural insecticide. This would be an insecticide found in a soil bacterium, Bacillus thuringiensis. This insecticide has been considered to be generally non-toxic to humans. It really targets insects, and only really a specific kind of insect. And if Bt corn is planted and helps protect the corn against corn borer or rootworm, then less pesticide would have to be applied in the field to protect the corn. So that's one potential environmental advantage of Bt, is reduced insecticide use. Another advantage is that without these corn borers boring holes through the corn plant that fungi then live in, without that, then there's actually reduced fungal toxins in this corn. At any rate, a huge amount of our corn crop is carries the Bt gene here in the United States, and an even bigger percentage of our cotton crop. As I said, cotton is very susceptible to insect attack, and so Bt cotton is a very important crop, not only in the United States but globally. Now, we've looked at some potential advantages of Bt, but there are some concerns too. Since the whole corn plant is genetically engineered, that means this Bt toxin is expressed in every leaf, in every cell, in the pollen, and we know that pollen from corn blows all over the place. It blows onto adjacent plants, such as milkweed plants that monarch butterflies exclusively eat, but the pollen also would blow over potentially to other fields, maybe organic fields, and so that's another concern. And here's a concern. There was a version of Bt that was considered so potent that it might be allergenic to humans. Not toxic to humans, but cause problems for humans. And so this Bt version put into corn, and the corn was called Starlink corn. Farmers were told, look, you can grow the Starlink corn, but it's not supposed to be for human consumption. This is strictly for animal feed. Well, many farmers didn't get that message, and it turned out that this particular form of Bt then was found in tortillas. It was found in human food derived from corn grown in the U.S. When they started to look, it was in every a huge shipment and silo. It was everywhere. And so critics have said, look, you know, what happened to our regulatory process here to protect people? Totally failed to keep this Bt out of our human food products. That's a potential problem. Now, herbicide resistance is another genetically modified trait that's very heavily used in agriculture. Herbicides such as Roundup will pretty much kill every plant, all the weeds. It would kill the agricultural plants, but if the agricultural plants have resistance to it, then they will survive and all of the weeds will be killed. So Roundup resistance then has been genetically engineered into soybeans, corn, canola, cotton, many crops. And it results in increased yields, cleaner fields. You can see that 91% of the soy crop in the U.S. is herbicide resistant, 70% of the cotton crop, 50% of the corn crop. But there are concerns about herbicide resistance too. Some people say, look, all we're doing is now encouraging more herbicide usage, which contaminates the groundwater, kills beneficial soil microbes. And then some crops, not maize or soybeans, but some crops like canola can cross-pollinate weeds. And so now if the weeds become herbicide resistant, people say, you know, that's not good. And then other people say, look, what's going on here? Farmers are now even more dependent on agrochemical companies. In fact, we've seen that chemical companies have bought up the big seed companies, and in order to market their herbicides along with resistant seeds. Now, in addition to these kinds of traits, which we're very familiar with here in Iowa and which have worldwide usage, there are many traits possible for disease resistance in just about any kind of crop. And disease resistance is a very important problem, especially in the developing world. So this is a potential application of genetic engineering which could solve problems that have not been solved before. In addition to those kinds of traits, of course, genetic engineering could be used to improve nutrition. And golden rice, that we've talked a little bit about already, is an example of that. We know that vitamin A deficiency is a big problem. Here we have the inventor of golden rice saying this rice could save 1 million kids in the developing world a year. Okay. Golden rice then incorporates a gene which allows it to synthesize carotenoids, which are yellow, producing the golden color, and those carotenoids are then precursors of vitamin A. And it was developed in order to give the seeds to the poor for free. But there are potential problems with this. People may not want a yellow-looking rice, and so golden rice hasn't really been planted in huge amounts. But there are other kinds of nutritional benefits that can be gained from genetic engineering. We know that soy is low in methionine. We know that maize is low in lysine. And of course, I work on quality protein maize. That is not a genetically modified trait, because in the developing world there is some resistance, at least by some people, to genetically modified. Another application of genetic engineering would be to put vaccines into crops that can be eaten, such as bananas. This would mean that the vaccine could be available to children without the need for refrigeration or all of the other infrastructure issues of developing or of delivering medicinal vaccine. And another idea is that maybe pharmaceutical products could be grown in crops, or other kinds of metabolic products. So we know that an AIDS vaccine is being developed to be produced in maize in Europe for use in the developing world. There are other proposals to turn crops which we know how to grow easily, can be harvested easily, and these products could easily be extracted from. But there are problems. If we're now putting pharmaceutical products in maize, how do we know that that maize won't end up in the food supply, just like Starlink corn did, right? Or even cross-pollination can be a problem. Now, genetically engineered animals are not yet approved for food, but here's some examples. There are transgenic fish, for example, salmon, that grow much faster than the normal salmon, but it's an environmental concern to produce these salmon. If they escape into the wild, they might outcompete natural species. And certainly transgenic animals, mammals especially, have been used for the production of pharmaceutical products. In milk production, pharmaceutical products that can be made in huge amounts, actually, compared to being made in bacteria. And so this is another application of genetic engineering. Now, getting back to the food supply, by 1999 then, with the advent of genetically engineered maize and soy in the United States, it turned out that 80% of processed foods in the United States could be seen to contain genetically modified ingredients. And when you go and look at ingredients lists of processed foods in the United States, we see a huge amount of corn and soy products. That's not the case in Europe. Okay? Their processed foods would not contain necessarily soy or corn ingredients, and so there has been a resistance to genetically modified in Europe, but in the United States, so much of our processed foods has these ingredients, and of course these crops are widely genetically modified. By 1999, the European Union said, oh, genetically modified is going to require labels, and in fact they blocked the import of genetically modified corn and beans. Now, by 2004 they lifted that ban, but there still is an anti-genetically modified sentiment in Europe. And one effect of that is that it affects African export crops, because a lot of crops in Africa are grown for export to Europe. So the European ideas about what's right and wrong with genetically modified then end up affecting what Africans who want to export to that market will produce. Also, by 1999, there were companies in the United States that said, well, maybe we'll just say we're not using genetically modified ingredients. Okay, so Gerber and Heinz went GM-free. McDonald's, a huge U.S. company, and Frito-Lay, actually, also a very big company. They own Pizza Hut, they own Pepsi. They went genetically modified free. And the USDA said, okay, if you're growing organic cannot be genetically modified. In 2002, Zambia just refused genetically modified maize as a food aid to help millions of people in a food shortage that year. The president of Zambia called genetically modified food poison. And again, you can see the influence of the European attitudes then on Africa. But genetically modified foods are continuing to be grown more and more acres or hectares around the world. So here we have places in the world where genetically modified crops are grown. You can see the United States, Mexico, much of South America, South Africa, some parts of Europe, India, China, Australia, etc. And there are genetically modified crops grown in Africa and many under development. Well, here we have a graph from 1996 to just the 10-year period to 2006 showing the growth in the adoption of genetically modified foods worldwide. Of course, the biggest part of this is the United States, Canada, and Australia, but Latin America has grown a huge number of GMOs. So by 2007, there were 300 million acres planted worldwide, 55% of that in the U.S. And the main crops, soy, corn, cotton, and canola. And cotton, especially in India and China. So this represents 12 million farmers. Some of them are very small farmers in developing countries growing cotton. Now, interestingly, in 2008, in related news, cloned animals were approved for human consumption by the FDA. Now, the goal in producing cloned animals is to produce high-quality meat and milk. The best animals having the best genetics are then replicated. That combination of genetics passed on by cloning. But these are not transgenic animals. That is, they don't contain DNA that has been manipulated in the test tube. They basically are taking a combination of genes from one animal and replicating that through the cellular cloning process to offspring. And as with all of the genetically modified products, a label is not required. I have to say, though, that cloned animals are not yet, or cloned food products are not yet in the stores. Now, there has been a controversy over biotech foods for decades now. And this controversy often pits consumer and ecology groups against the multinational corporations. So the consumer groups are saying, we should know what it is we're eating. We should have a say. Things should be labeled. The ecology groups are saying these foods should be safe for the environment. The multinational corporations are saying we're delivering great stuff, and we shouldn't be unfairly regulated. And there's a lot of people involved, farmers, scientists, government agencies, who may actually sort of be caught in the middle of this controversy. Paul Thompson is a philosopher, of course, been to the Iowa State campus, wrote a book on ethical considerations of biotechnology, and his arguments are that genetically engineered foods have a lot of positive potentials to increase productivity, purity, safety, nutrition, food quality, and actually also could improve sustainability and benefit the ecosystem. He argues that this process is not inherently harmful, that at the molecular level it's really similar to plant and animal breeding, and that you would expect that the outcome would be beneficial unless, of course, the technology were misused, and any technology can be misused. So this is a powerful technology that could help humanity, according to this argument, and not only that, but in our legal and regulatory environment, bad ideas are going to be weeded out by the market, by regulation, by lawsuit. Take Starlink corn, for example. It caused such a problem. I don't think that farmers are going to plant Starlink corn. They don't want to be sued. Okay? So our regulatory and legal system helps to weed out bad ideas. That's the argument. Now, there are arguments against genetically engineered foods, and there's many different kinds of parts. One of the arguments is, look, maybe there is a food safety risk. We don't really know. If you look at the history of science, there's always been innovation, and people tout these scientific discoveries as being great, and then 20 years later we find out that they were extremely harmful. Radiation, for example. People used to be able to walk into a shoe store and put their foot in an X-ray machine and look at their bones. It was great technology. Of course, we know how damaging that technology is now after the fact. And this argument says genetic engineering might be the same way. 20 years from now, we might discover that there was a safety downside. So we need to go very slow. There's, of course, the argument that there's a safety risk for the environment. You begin genetically modifying all of these plants or animals, and they can escape or breed or pollinate in the environment and spread. Then there's arguments, consumers especially saying, look, we would like to see labels. Why can't we see labels that tell us that things are genetically modified, and then we can make an informed decision about them? Other people say, look, genetic modification is not natural. It's playing God. We should not be doing that. Now, for this particular argument, I'd like you to consider, though, what natural means. What does natural mean? And the reason I bring that up is that for 10,000 years, humanity has been eating agricultural products. That is, the domesticated plants and animals. And domesticated plants and animals are different genetically. They meet human needs much better. So are they natural? So when you use this argument that genetic engineering is not natural, then you need to be careful about defining and discussing what is natural. If you say that any kind of genetic tampering with nature is unnatural, then you would have to throw out all of agriculture. Another argument is that genetic engineering, who does it really benefit? It benefits the big multinational companies, but it doesn't really benefit consumers, and it doesn't benefit developing nations. So let's look at some of these arguments. The unintended consequences argument basically says there might be a food safety problem. These genes are inserted into the genome randomly. Do we really know what they're going to do there? Are they going to produce allergens or new gene products? And I would like to say that there really is no evidence of GMO food safety problems to date. But food allergens are a very important issue. 90% of food allergies come from these foods: eggs, fish, shellfish, milk, peanuts, soybeans, tree nuts, and wheat. So these are not good sources of genes for genetic engineering, because of course you might be introducing the allergen into another food, and genetically modified foods do avoid genes from those sources. What about the argument for labeling? Well, it says, look, genetically modified foods might look like other foods, but they're not really equivalent. They're not really equivalent because of the process that's been used in genetic manipulation. And so we should use the precautionary principle. We ought to go slow. And, you know, consumers are savvy enough to want to know and to be able to appreciate the process used. Generally, I think food manufacturers are concerned about any label about the process used because they don't know how sophisticated consumers might be. But there are consumers who say, we want to know what process is used. An example is, let's say, decaffeinated coffee. There's a couple of different processes. One that uses organic chemicals, that is chemicals that are based on organic solvents to extract the caffeine, and some of those organic solvents might remain as a residue in the coffee. On the other hand, there is an aqueous or water-based extraction method which would not be leaving these chemicals in the coffee. And those two different processes, consumers say, we can distinguish between those, and we want to be able to buy our decaf coffee based on a choice. Likewise, with genetic modification, we want to know if our food is genetically modified. We have a right to know and choose. But the arguments against labeling are these, that labels are important, but they're most important if they indicate hazards or known problems. If you put on a label that doesn't really suggest a hazard, that might be misunderstood by people. So the argument is that a label is going to suggest that there's a problem. And of course, the FDA and the food industry contends that there is no problem with genetically modified foods. Of course, it's expensive to change labels, and the FDA says, look, we're labeling the things that are important to label. We make sure that there's a label change if there's a food allergenicity change, if there's a change in nutrition, if there's a change in food quality. But genetic modification is not one of those things that causes a significant change. And then here's another question. Will genetically modified crops feed the world? Norman Borlaug, among many others, would argue yes. And here's some of the arguments. These crops are size neutral. In other words, you don't have to have a giant farming operation to use genetically modified seeds. Small growers could benefit. In many cases, they may reduce the inputs. For example, if you're using BT corn, there's a reduced input of a pesticide needed, right? So that lowers costs. Of course, there are increased yields, many traits like disease resistance, reduction of weeds, and weeds are a huge problem in the developing world. So when you use herbicide-resistant crops, the fields are very clean. And all of that is going to result in increased profits. If it's increased profits for the small farmer, then it's good for the poor and will help feed the world. That's the argument. But the argument against genetically modified crops feeding the world basically is this, that we have these crops because biotech companies are trying to make a profit. They're targeting the wealthy to do that. Intellectual property is expensive. So what's really needed is public research in developing countries to develop genetically modified crops, and that is actually occurring. Here we have the International Center for Genetic Engineering and Biotechnology in India, and there are many countries that are pursuing genetically modified research to develop crops for their country. So it isn't like these countries are depending on the United States or even necessarily on biotech companies for these seeds. But this argument would go on to say that because in many cases these seeds are expensive when they come from biotech companies, of course the poor can't compete, so they're driven from the land and we get more undernutrition. The same thing that happened in the Green Revolution, right? The argument would say instead of biotech seeds, what the poor really need is land. They need their own land. They need access to water. They need roads. They need education. They need credit. All of these things are much higher priorities than genetic modification, which really, in the end, if it comes from a biotech company, is going to help a few wealthy and probably actually increase hunger. And then finally, another argument, the same argument used against the Green Revolution, is that what kind of agriculture is this biotech agriculture? It's the same unsustainable commercial or industrial agriculture, right? Based on monoculture with erosion, fertilizer and pesticide runoff, and generally requiring a lot of fossil fuels to produce these crops, all of which is not good for feeding the world in the long term. The American Corn Growers Association kind of sums up a part of this issue. They say, while feeding the hungry is a laudable goal, current record feed stocks in the U.S. is still not finding its way to those who need it the most. Therefore, the real reasons for hunger is not necessarily the lack of food, but the lack of income to purchase and the absence of an infrastructure to get to the food to those who need it the most. If the hungry cannot be fed with current worldwide overproduction, what guarantee is there that additional productivity will solve the problem?